Hello, my name is Katrina and I work in clinical development at Ionis Pharmaceuticals Inc. And I'm delighted to be able to talk to you today and provide you with an update on our Alexander disease program. So I'm going to begin today by telling you a little bit about Ionis, a little bit about antisense oligonucleotides, and then provide you with an update on our Alexander disease program. Ionis was founded in 1989 as Asos Pharmaceuticals. We changed our name around 6 years ago, I think you can imagine why, to Ionis. We're located in Carlsbad, sunny California, and we're the leading innovator in RNA-targeted therapeutics, which I'll talk to you more about in a moment. Ionis is made up of world-class scientists and leaders who have a passion to deliver transformational medicines to the patients who need them. Ionis is largely focused on the research and the development of antisense medicines, which are designed to disrupt the disease process. We have a number of different drug programs in development with 3 recently approved medicines: Spinraza or nusinersen for SMA, and Tegsedi and Waylivra for 2 other rare indications. We're really excited to test GFAP targeting antisense oligonucleotides, or ASOs, as a possible treatment to Alexander disease. And I wanted to talk to you briefly about how ASOs work. And so to do this, the first place that we need to start is to think about how proteins are made, which starts with DNA, which you can see here pictured on the left. The DNA is the genetic material in your cells. You can think of DNA as the master cookbook to make all the proteins your body needs. Each protein has its own unique recipe called a gene, and DNA is the cookbook for all of those genes. Your cells are able to make millions of different proteins, and proteins are tiny molecules that are required for nearly everything related to how cells live and work. For example, they serve as structural building blocks, they're responsible for waste cleanup and routine maintenance, they receive signals, they send signals, they mobilize responses to viruses. Every cell in your body contains tens of millions of proteins at any given time, and they're all working in their own way to keep you healthy. So we're heavily dependent on our ability to make the proteins we need in the quantity we need when we need them. DNA naturally exists in a tight double helix structure in a special compartment of the cells called the nucleus. Being double-stranded and protected in the nucleus helps to keep it safe. After all, it's the master cookbook, so it's important that the DNA is well protected. When your body needs a protein, the DNA unwinds a little bit to expose the recipe for that particular protein, remember, also called the gene. And here's where the really interesting thing happens. A temporary copy of the recipe is made, and the copy is called mRNA, which stands for messenger RNA. The mRNA is much more mobile than DNA. This mRNA copy of the recipe is able to leave the nucleus and interact with the cellular machines that can read the mRNA and use that information to produce a protein. So that's how a GFAP protein is made. There is a tiny area in the DNA that holds the recipe for making GFAP. When your body wants to make GFAP, it takes a copy of the recipe, the copy is called mRNA, and the mRNA recipe for GFAP goes off to the cellular machines, which then produce the GFAP protein. And so Ionis develops a technology called antisense oligonucleotide, or ASOs, and this type of drug acts on RNA to affect the amounts of protein that are made. So let me show you now how this works. So here is the same process that I just showed you. We have DNA at the top, then an mRNA copy is made, and it's the mRNA that is destined to leave the nucleus to be used to produce a protein. Now, ASOs can be designed to work in a dozen of different ways. And on this slide, I'll show you the most well-studied ASO mechanism. This is a way to use an ASO to lower the amount of a particular protein that's made. This also happens to be the ASO mechanism that we're using in our Alexander disease program. So what we do with antisense oligonucleotides is we design this little molecule shown here by the purple strand. The purple molecule is made of chemically modified DNA. We design the ASO so that it will recognize only the mRNA of interest. When the ASO binds to the mRNA, it creates this duplex, which is shown here by the orange and purple strands interacting with each other. Now, our bodies are designed to recognize errors in genetic material. Double-stranded DNA doesn't raise any alarms. After all, that's exactly how you would expect DNA to be, two strands interacting with each other. It turns out that double-stranded RNA also doesn't raise any alarms. RNA is usually a single strand floating around, but there are situations where two strands of RNA will interact with each other, and so there's nothing wrong with that. But when RNA binds to something that looks like DNA, which is what we have with this orange and purple mRNA ASO duplex, the alarm sound. RNA should never interact with DNA. Now, your body recognizes this error, and it turns out that your body has this special built-in mechanism to deal with this error. And this is a naturally occurring enzyme called RNase H1. And it finds the RNA DNA duplex and it destroys it. Specifically, the RNase H1 enzyme cuts the mRNA, and after the mRNA has been cut, it can't be used to make a protein. So with our ASOs, we're really tricking the body into cutting up the mRNA message, which should result in less protein being made. As I note here on the right of this slide, ASOs are highly specific and titratable. By highly specific, I mean the ASO is designed to interact with a particular mRNA. For Alexander disease, we're interested in lowering the amount of the GFAP protein that is made. So we design an ASO that will interact only with the GFAP mRNA. We don't want it to interact with the mRNAs for any other proteins because we don't want to change the amount of any other proteins. Our singular interest is in lowering the GFAP protein. By titratable, I mean that if we administer small amounts of ASOs, there will be small lowering in the amount of protein. And if we administer a large amount of ASOs, it will cause a larger magnitude change in the amount of protein. One thing that's important to understand is that ASOs can be treatments, but they're not cures. Like any other drug, the ASO will eventually be cleared from the body. So the ASO's effect on protein levels will only last for as long as the ASO stays in the body. So let's talk now about the possibility of an ASO treatment for Alexander disease. So in Alexander disease, cells make too much of a particular protein called GFAP protein. Everybody has 2 copies of GFAP, just as you have 2 copies of all of your genes. In the case of Alexander disease, there is a little change in the DNA from one of those copies. We call this change a mutation in the gene. When you have the mutation for GFAP, it results in overexpression of the gene, which means that too much GFAP protein is being made. So here, we've designed an ASO that will target the GFAP mRNA, and using the mechanism that I just showed you on the last slide, the ASO binds with the mRNA, which causes it to be degraded. If the GFAP mRNA is being degraded, it cannot be used to make the GFAP protein. So we would like to use this approach to treat individuals with Alexander disease. The underlying cause of the disease is a mutation of the GFAP gene on the DNA, and that mutation leads to too much GFAP protein. Remember, ASOs don't change DNA, so the mutation will remain in the DNA, but we would like to administer an ASO that intercepts the GFAP mRNA and leads to its destruction, which should counteract the mutation by lowering the production of the GFAP protein. So here, I want to summarize for you our Alexander disease clinical trial design and where we are in the study. I think one important thing to remember when we talk about clinical trials is that they're research. They're experiments. And when we design a clinical trial, we have a number of questions that we're looking to answer, such as, is this particular ASO safe? Is it beneficial? Do people with Alexander disease benefit from taking our medicine? So we've designed an experiment to generate data to help answer these questions. Another important thing to think of is that really we're at the beginning and not the end of our experiment. we're at the stage where we can learn about the possibility of an ASO treatment for Alexander disease. So in our clinical trial, we're obviously enrolling patients with Alexander disease. And this is important because sometimes the first trial of a new drug is in healthy volunteers, but that's not the case here. We're going straight into individuals with the disease. Study participants receive the drug every 12 weeks, and participants will either receive 10 doses of ION373, which is the name of the drug, or they receive 5 doses of placebo, followed by 5 doses of ION373. Patients, parents, staff, and us here at Ionis are masked to the treatment assignments. The study drug is administered intrathecally into the CSF via lumbar puncture. We have a broad age range from 2 to 65 years, and we're asking that patients be ambulatory and if they are 5 years or older. And importantly, we require that patients be able to safely participate. In this study, we have endpoints in the trial that include performance tests, questionnaires, physician reports, lab tests, and so on. So it's a long set of assessments, but at the end of the study, we'll use the tests that are administered to measure changes in motor function, communication, cognition, and everyday functioning, and also assess the safety and tolerability of the drug. We have several clinical sites already identified around the world participating in the trial, and we're aiming for approximately 42 participants. If you'd like to learn more about the Alexander disease program, you can visit our website at ionistrials.com. Clinicaltrials.gov is also a great resource for information. You just need to search the term shown here, and you can also always reach out to our patient group here at Ionis by emailing or calling. Thank you so much for listening. I'd like to give you the opportunity now to ask us any questions.